Hello, my name is Lily Yellen, and I'm a team member of Connections Official, a company that specializes in genealogy research services. Today, I will cover the basics of the mitochondria structure and function, mitochondrial DNA, theories of maternal inheritance, mtDNA testing, and its advantages and disadvantages. In relation to the structure of the mitochondrion, which is the singular of mitochondria, is a rod-shaped structure found in the cytoplasm of all eukaryotic cells. There are hundreds to thousands of mitochondria in each cell. And the most important function of the mitochondria is the production of energy to support all cell functions. This is accomplished via the production of adenosine triphosphate, which is the main source of energy of the cell. And this is accomplished via a process of oxidative phosphorylation. Relative to the size of other organelles, mitochondria are small structures between 0.5 and 1 micrometer in diameter. The structure comprises an outer membrane, an inner membrane, and a gel-like material called the matrix. The outer membrane and the inner membrane are made of proteins and phospholipid layers separated by the intermembrane space. The outer membrane has a large number of special proteins known as porins, which make it permeable to major metabolites and ions, and play an important role in ATP regulation. The inner membrane of mitochondria is a fairly complex structure. It has many folds that form a layered structure called crista, which means ridges, and they help in increasing the surface area inside the organelle. The mitochondrial matrix is a viscous fluid that contains a mixture of enzymes and proteins. It also comprises ribosomes, inorganic ions, mitochondrial DNA, nucleotide cofactors, and organic molecules. In addition to nuclear DNA, we also have mitochondrial DNA. And as the name suggests, it is located within the mitochondria. Mitochondrial DNA constitutes a minuscule portion of the human genome, which comprises 30 billion nucleotide base pairs, or about 30,000 genes. For comparison purposes, chromosome 1, which is the largest chromosome, contains roughly 249 million nucleotide base pairs and accounts for about 8% of the human genome, while the Y chromosome, which is the second smallest chromosome, contains about 58 million base pairs, translated roughly into 70 to 200 genes. In contrast, mtDNA is comprised by only 35 genes and accounts for 1% of the human genome. On the image on the left, we can view the typical nucleus encoded DNA wrapped around histones, which is tightly packed into chromosomes. And as it unravels, we can appreciate the characteristic linear structure, which resembles a twisted ladder held together by 4 nucleotide base pairs: adenine, cytosine, guanine, and thymine. On the other hand, the mitochondrial genome is a circular, double-stranded DNA molecule, and it lacks histones. As discussed before, mitochondrial DNA is located outside the cell nucleus in the cytosol. And unlike autosomal chromosomes that are only present in the nucleus, one pair at a time, there are multiple copies of mtDNA present within each mitochondrion. The mitochondrial genome encompasses a total of 37 genes, as mentioned before. Of the 37 genes, there are 13 protein encoding genes, as well as 22 transfer RNA genes and 2 ribosomal RNA genes involved in the translation of protein. Encoding of mitochondrial proteins involve both the mitochondrial and nuclear genomes, and it works more or less like this. The 13 protein encoding genes encode roughly 100 mitochondrial proteins. Nonetheless, mitochondria require nearly 1,000 different proteins to perform all their complex functions. Therefore, it is necessary for the remaining 900 proteins to be encoded by the nuclear genome. The nuclear genes are translated and transcribed in the cytosol, and proteins imported into the mitochondria combine with the polypeptides made within the mitochondrial matrix from mitochondrial DNA. Any disruption of this concerted interaction between the two genomes may cause cell dysfunction and disease. So in the human sperm, there are approximately 50 to 75 mitochondria, all coiled around the tail, and they are arranged end to end in the midpiece region. Mitochondria have a high capacity to produce reactive oxygen species. Therefore, mtDNA is more vulnerable to mutations caused by free radicals than nuclear DNA. There are several factors, including the lack of histones, the absence of cytoplasm, and the limited production of antioxidants that make mitochondria highly susceptible to peroxidative damage. The oxidative stress induced by free radicals can cause degradation of the sperm's plasma membrane, fragmentation of the mitochondrial DNA, and dysregulation in the sperm transcribed messenger RNA. So this is where the imperative for uniparental mtDNA heritage comes in, to avoid defects in reproduction through the elimination of possibly mutated mtDNA. Not only is the degradation of paternal mtDNA essential to maintaining the welfare and integrity of the embryo, but also to prevent mitochondrial diseases. Thus, single-sided mtDNA inheritance through the mother ensures all mtDNA copies in the offspring are identical to hers, and this is referred to as homoplasmy. So there are at least two theories that have been proposed to explain the maternal inheritance of mtDNA. One is the degradation of paternal mitochondrial DNA via an active process such as mitophagy or endocytic degradation. And the other one is the dilution of paternal mitochondria via passive dilution. In this case, paternal mitochondria are diluted in the cells of the embryo during cell division until they become undetectable. Mitophagy or autophagy is a mechanism through which dysfunctional mitochondria are degraded to ensure cellular homeostasis. In other words, that only healthy mitochondria survive to ensure cell processes are properly maintained. Presumably, this mechanism evolved to enable somatic cells to eliminate damaged mitochondria in conditions of oxidative stress, nutrient deprivation, and aging, as well as to remove mitochondria during normal cell differentiation. There are different pathways through which mitophagy can occur. We can see three of those pathways on this slide. But for the purposes of this presentation, let's focus on the first pathway. There are certain proteins like PINK1, which is a protein kinase, which are thought to bind to damaged mitochondria to induce autophagy of this organelle. PINK1 shares a common pathway with the E3 ubiquitin ligase parkin, and the interaction between the two works as follows. As accumulation of PINK1 on the outer membrane of damaged mitochondria occurs, PINK1 phosphorylates ubiquitin, which in turn triggers recruitment of parkin from the cytosol to the mitochondria and activates its E3 ligase activity. At the same time, ubiquitin molecules mark the mitochondria for degradation and initiate the formation of autophagosomes. And next, the autophagosomes engulf the mitochondria, which are then digested by lysosomal enzymes. The mitochondria of a spermatozoa can be ubiquitinated since they are in the testes before fertilization. However, after fertilization, the signal for selective destruction is amplified, and this allows for its recognition by the oocyte degradation machinery. There are additional pathways that are ubiquitin independent, like the next two below the pink one parking pathway, but the basic principles for the signaling of mitochondrial destruction are similar. So paternal mitochondrial elimination may also involve the endocytic pathway. In this case, the sperm tail content may be too large to be engulfed by an autophagosome. Thus, microvesicles from the oocyte cause an initial tail fragmentation. So the autophagy system proceeds with degradation. Ubiquitination also plays an important role in signaling microvesicles, participating in the initial degradation of the spermatozoid's tail. Lastly, another hypothesis for maternal inheritance of mtDNA is passive dilution. It is believed that after oocyte fertilization by the sperm, the cell division of the embryo will dilute the sperm mitochondria into the maternal medium, since the latter are in greater quantity when compared to the paternal ones. As a result of all of this degradation, mitochondrial DNA is inherited almost invariably from a mother to her children every time, generation after generation. Although mitochondrial DNA from the father has been detected extremely rarely in humans, systematic research studies have failed to yield any indication of paternal inheritance. Therefore, at present, maternal inheritance of mtDNA in humans can be regarded as the rule. Thus, inheritance of mtDNA stops with men, and the uniparental line from mother to children continues through the generations. So from a genealogical perspective, there is an advantage to testing mtDNA. The reason is that this maternal mode of inheritance is one that enables researchers to trace related lineages back through time, highlighting the maternal ancestry of a population without confounding effects of biparental inheritance and without the recombination that is inherent in nuclear DNA. So how is mitochondrial DNA tested in genetic genealogy? As you may recall from previous slides, the structure of the mtDNA molecule contains a control region and a large coding region. So in the control region, there are two hypervariable regions, 1 and 2. All of these elements are tested for genetic testing of individuals. The hypervariable regions are likely to change from one person to the next unless people are descendants from the same maternal line. On the other hand, the coding region is slower in mutating than the control region, and mutations can be used to define haplogroups. Now, there are two different tests to identify different mtDNA regions. One is mtDNA sequencing, and the other one is single nucleotide polymorphism testing, or SNP testing. So mtDNA sequencing tests analyze either all or a portion of the mitochondrial DNA. mtDNA sequencing tests include low-resolution tests and high-resolution tests. The low-resolution tests sequence only the hypervariable regions, whereas the high-resolution test additionally sequence the coding region. The full sequence testing is recommended over the low-resolution test for genealogical purposes, obviously because they provide more information about the ancestry of the person, of the tester. SNP testing involves the analysis of pieces of DNA that can vary from person to person, but individuals who are closely related should have the same SNP at every location or most locations tested. SNP testing will test hundreds or thousands of SNPs around the circular mtDNA rather than examine the entire genome. So let's take a look at some of the advantages of mtDNA testing in genealogy. There is very little recombination or changing of maternal mtDNA as it passes down through the generations. Therefore, mtDNA molecules in the offspring are identical to those of the mother. Test results can be matched to the results of other testers, which affords people the opportunity to discover distant relatives who share direct matrilineal ancestors. Autosomal DNA tests can identify varied haplogroup defining locations. Nevertheless, they cannot detect every location needed for a complete haplogroup, which is necessary to discern the history of someone's ancestors. Mitochondrial DNA testing allows a tester to peer behind the veil of a brick wall in a specific line to elucidate where his or her ancestors originated hundreds or thousands of years before surnames emerged. Test results, including mutations, may hold interesting information, such as genetic locations where there are insertions or deletions unique to a tester and his or her potential matches. And some of the limitations of mtDNA testing are that since mtDNA is only passed from mother to children, mtDNA tests ignore the father's side of the family. It ignores also the line of the mother's father, as she only received mtDNA from her mother. Tests also will miss any relatives outside of the direct maternal line. Tests may be able to provide information about distant relatives, but only about a subset of the family. Thank you for listening to this presentation. Please visit Connections Official at www.connectionsofficial.com for additional information on genetic testing.